Hello, my name is Tomoyo Yoshida from the University of Tokyo. Today I will present our study titled Multimodal Single-Cell Profiling of Master Immune Infiltrates and Spatial Transcriptomics Reveal Shared and Disease-Specific Programs in Idiopathic Inflammatory Myopathies. Idiopathic Inflammatory Myopathies, or IIM, are systematic autoimmune diseases characterized by muscle inflammation and immune cell infiltration. While previous studies have identified features such as type 1 interferon signatures, the cellular heterogeneity of immune infiltrates in muscle tissue remains poorly understood. To address this, we performed a multimodal single-cell analysis of muscle immune cells combined with spatial transcriptomics. We obtained muscle biopsies from 18 patients with IIM, including dermatomyositis and antisynthetase syndrome. Matched peripheral blood samples were available for 5 patients. CD45 positive immune cells were sorted and analyzed using single-cell RNA sequencing with surface protein profiling and paired TCR-BCR sequencing. In addition, spatial transcriptomics using the Visium platform was performed in 2 antisynthetase syndrome samples to connect immune cell populations with tissue pathology. In total, we analyzed more than 43,000 immune cells derived from muscle and blood. First, we defined the overall immune landscape of muscle infiltrating cells. Across samples, immune cells were broadly classified into T cells, B cells, NK cells, monocytes, and dendritic cells. Interestingly, hierarchical clustering based on cell type composition revealed that dermatomyositis cases were predominantly NK cell rich, whereas antisynthetase syndrome showed greater heterogeneity. Next, we analyzed CD8 positive T cells. Subclustering revealed a population of granzyme K high tissue resident memory T cells characterized by CD69 and CD103 expression. These cells were markedly enriched in muscle, expressed interferon gamma, and showed clonal overlap with proliferating CD8 T cells, suggesting local expansion within the tissue. These cells showed limited variation across IIM subtypes. Therefore, granzyme K high CD8 resident memory T cells represent a shared immunological feature across IIM subtypes and appear to be a major source of interferon gamma in muscle. We also observed expansion of CD4 helper T cell subsets, particularly Th1 and TPH-TFH cells in muscle. These cells showed increased clonality and CXCR13 expression, suggesting local T-B cell interaction within inflamed tissue. We next examined differences between clinical subtypes. In antisynthetase syndrome, we observed expansion of cytotoxic CD8 tumor cells, which showed strong expression of cytotoxic genes such as granzyme B and perforin, and exhibited prominent clonal expansion. In contrast, MI2 positive dermatomyositis was characterized by abundance of CD56 bright NK cells in muscle. Cell-cell communication analysis suggested that these NK cells may recruit type 1 conventional dendritic cells via the XCR2, XCR1 signaling axis. We also identified mate cells with strong interferon-stimulated gene signatures enriched in MI2 positive dermatomyositis. Together, these findings highlight distinct immune programs between IIM subtypes. Finally, using spatial transcriptomics, we examine how these immune processes relate to tissue pathology. We identified spatial gene modules corresponding to interferon responses, T-B cell interaction, and muscle repair processes. Notably, regions enriched for T cells and B cells overlapped with interferon-inducible areas and regions of muscle damage, indicating that immune activation and interferon signaling converge at sites of tissue injury. In summary, our multimodal single-cell and spatial transcriptomic analysis revealed both shared and disease-specific immune programs in IIM. These findings provide a multidimensional view of immune processes in inflamed muscle and may help refine our understanding of disease heterogeneity in idiopathic inflammatory myopathies. Thank you very much for your attention.